I will explain a bit about what this title means to those of you who might not be familiar, but I'm going to talk about generally non-coding exons in a rare disease context. So first, just to define rare disease. So in the UK, we define a rare disease as a disease that affects fewer than 1 in every 2,000 individuals. This definition varies across the world, but the general mantra around rare diseases, although they're individually rare, they're collectively incredibly common and affect about 1 in every 17 individuals. So around 70% of them start in childhood, and over 80% are thought to have a genetic basis. And actually figuring out what that genetic basis is, so finding the DNA mutation that is causing a disease in a patient, is incredibly important for patient and family management. So sometimes it's the first diagnosis the patient gets, so we don't know what's wrong, we don't know what's causing their disease, and actually genetic mutation is what tells us. It allows us to screen additional family members. So sometimes if you have an individual with a mutation who develops a disease, say, later in life, we can then find other individuals in the family that are also at risk of, say, a cardiovascular phenotype, for example, or it can help us with prenatal screening and recurrence risk counselling. It can also help us to inform how a disease is going to progress and how a patient is going to do clinically, and increasingly it leads towards us actually finding personalised treatments, so treatments that are geared towards correcting or targeted towards the specific mutation that we find in a patient. But currently, although over 80% of diseases are thought to be a genetic in basis, for the majority of rare diseases, our diagnostic rate is very low. So we actually find a genetic cause in around 40% of individuals. Now, this varies by phenotype and gene. This is a plot from the Deciphering Developmental Disorders cohort based in the UK from this paper from the New England Journal of Medicine, where across their cohort of a broad range of developmental disorders, their diagnostic rate is about 40%. But the limitation in our current clinical genetic testing is that we focus very heavily in the around 1.5% of the genome that directly encodes protein. Now, this is a really sensible strategy. We can find 40% of diagnoses in 1.5% of the genome. Why would we not look in the protein coding regions first? But given we've kind of stagnated this, we can look in other places. Now, my talk was titled about the non-coding exome, so I kind of need to mention what I'm talking about here. So to some people, they think that the exome is synonymous with protein coding. So they think every exon in the genome encodes protein. But this is actually incorrect. So an exon is actually any region of the genome that is transcribed into RNA and actually retained in the transcript after splicing. So on the top here, I've got a protein coding gene. I'm pressing the wrong button, where these coding exons are shown with these green boxes. But actually, in a protein coded gene, we also have these untranslated regions, both 5' and 3' to the coding sequence. And when after splicing has happened, we have these some exons that are solely UTR. So not all of them are protein coding. We also have an abundance in the human genome of non-coding RNAs, and they also have exons and introns, but none of these are protein coding. And actually, protein coding is not the rule. There are actually more exonic sequence in humans that is not protein coding than is protein coding. So what I'm showing you here is different annotations. So coding sequence, untranslated regions, non-coding RNA, and other, which is things like pseudogenes. And the height of each of these bars is the proportion of the genomic, well, the amount of genomic sequence that is in humans accounted for that annotation. So as you can see, actually, the most abundant type of exonic sequence are these non-coding RNA species. This actually varies a lot through evolution. So more basic organisms actually have much fewer of these non-coding RNAs and much shorter UTRs, and much more of their genome is due to protein coding sequence. And what the coloured in bits of these bars represents is quite often in a clinical setting, we use what we like to refer to as whole genome sequencing to look for whole exome sequencing to look for diagnoses. And the coloured in proportion of these bars is the proportion of that exonic sequence that is captured by this whole exome sequencing approach. So basically, my lesson here is we shouldn't use the word whole. It is protein coding exon sequencing because we're capturing about 95% of the coding exome, but pretty much nothing of anything else. So in the UK, we're incredibly fortunate that quite a few years ago now, the UK government decided to invest in genome sequencing. And it's actually part of our strategy to actually use genome sequencing as a first-tier test for individuals with rare disease now. So anybody that presents in the clinic with a developmental disorder is immediately given genome sequencing rather than starting with looking at specific genes or indeed using an exome. So we now have a lot of data, and this gives us the opportunity for my interest here to look at potential diagnoses in these non-coding exons. So in this talk, I'm going to touch on two of these different types of non-coding exons, and I'm going to start with a really cool recent story of a non-coding RNA. So this is a paper that came out in July, where we looked at a specific gene, RNU4-2, which is a small nuclear non-coding RNA gene. And we found that variants in this gene are an incredibly frequent cause of neurodevelopmental disorders. So just to start with saying that this was a cast of over 100 people that contributed to this work. They were all amazing. It was a huge collaboration between scientists and also clinical teams across the world. So UK, mainland Europe, also Australia and the US all represented in here. So this started by an awesome PhD student in the team, Yuyang Chen, who was looking at non-coding RNAs in the Genomics England 100,000 Genomes project dataset. So this is a flagship project by Genomics England, who have collated all of this whole genome sequencing data that's been done in the UK. And the first project was to do 100,000 genomes of individuals with rare disease or cancer and their families. So this is a project that's now completed, but the data is all available within a secure research environment. So Yuyang was looking at this data, and he came across a really interesting variant. It's a single base insertion in the center of the RNU4-2 gene. And we found this in 46 individuals in Genomics England. And actually in 40 of these, we knew that it had occurred de novo. That is that we have two unaffected parents that don't have the mutation, and the mutation has newly arisen in the affected child. Now, Yuyang showed this to me and I said, well, it's clearly complete rubbish. That's clearly a sequencing error. Something's going wrong here. So de novo mutations are random events. We don't expect to see this exact same de novo mutation in a large number of individuals. Our occurrence is normally maximum of 2 to 3 individuals, unless there's some kind of germline selection going on that means that a single mutation is driven to high levels. But I was very suspicious because also this is an insertion variant, which we know sequencing quality can be a little bit more suspect. So I said, well, this is clearly rubbish. Please, let's move on and put a filter in there for a maximum allele count. And we'll look at things that are a bit more rarer than this. But Yuyang, thankfully, was a little bit persistent. He said, but hang on a minute, all of these have an undiagnosed neurodevelopmental disorder. And it actually comprised about 0.5% of the nearly 9,000 probands that we have in this cohort. So we started digging into this a little bit deeper. So we did a lot of work trying to check that this wasn't just a sequencing error, but the genotype quality looks good. We have about 50% of the reads being reference versus alternate. So this looks like true heterozygous calls. And the coverage, the number of reads covering the position was very good. Susan Walker is an absolute superstar that works at Genomics England. She went through painstakingly the IGV plot, so looked at the individual reads covering the variant position in each child and both their parents, and saw that all these variants looked like they were good quality calls. So we started looking for this variant elsewhere. So although we found it in 0.5% of these undiagnosed neurodevelopmental disorder patients, it wasn't in any of the NDD probands that had been diagnosed. It wasn't in any individuals that didn't have an NDD phenotype. Sorry, NDD, I mean for neurodevelopmental disorder. It wasn't in any of 33,000 unaffected individuals. These are mainly parents of individuals in the cohort. It wasn't in the NOMAD population sequencing cohort. It wasn't in the 250,000 individuals from the US-based All of Us sequencing cohort. And it was in a single annoying individual in the UK Biobank. We don't really understand this finding yet, but this individual has a ICD code that says personal history of diseases of the nervous system and sense organs, which we don't know what that means. But despite that, there is this incredible enrichment where we see this variant in 46 of these Genomics England individuals and only a single individual of the nearly 500,000 individuals in the UK Biobank. So looking at this variant, so just to orientate you to this plot, on the top here is de novo variants found in Genomics England. And on the bottom here are variants found in the UK Biobank in those 500,000 individuals. Each dot is a variant, where the length of the string on the lollipop is the number of individuals that carry that variant. So on the bottom here, the longer the string, the further down the dot is, the more individuals carry the variant. This is the whole sequence of the RNA. It's 141 base pairs long. It's very short. And in the middle of this region here, highlighted in red, we see a depletion of variants in the general population. So none of these lollipops are very long, and there's actually gaps, although there isn't across the rest of the sequence. And it's in this depleted region where this tower of triangles, this is our variant that we identified, is found. So given we saw this depletion, we decided to look in this wider region. And actually, we find multiple other variants in this region that are all individuals with undiagnosed neurodevelopmental disorders. The majority of these individuals have single base insertions, although there are also a few single nucleotide variants, so base changes in here. So we then worked with Anna de la Lauria, who's at the Broad Institute, to look in a whole host of different cohorts to find additional individuals with variants in this region. And this, incredibly quickly, we ended up with a cohort of around 115 individuals. These variants are everywhere. They pop up in every single cohort of whole genome sequencing that we look at. And again, this kind of single base insertion that we initially identified is actually a predominant mutation and is in 75, well, about 3/4 of the cohort. So Anne and her team, specifically Gabrielle and Sarah, looked into the phenotype associated with variants in this region. So in total, we had 49 of these individuals where we had deep phenotype information, where we could contact clinicians or recruiting centers. And 100% of these individuals have global developmental delay, with the majority of it being marked as severe. And they also have intellectual disability. And a large proportion of the cohort have microcephaly, are short in stature, have speech abnormalities, with most of them being completely nonverbal. They also have hypertonia, vision issues, difficulties feeding, and most of them also have seizures. Most of these individuals have involvement across multiple organ systems, so it's not specifically a neurodevelopmental syndrome. And one of the interesting things that we're starting to see, although we don't have a huge amount of data on this, because we only have 4 of these 49 individuals with these single nucleotide variants, they look to be associated with a milder phenotype than these single base insertions. So actually, these individuals all have moderate rather than severe developmental delay, and the only individuals that have fluent speech have these single nucleotide variants. There is also, apparently, if there are any dysmorphologists in the room, a distinct facial dysmorphism associated with this syndrome. I'm not very good at looking at faces, so I can't see it, but some of you might. So one of the things that was most striking about this discovery is its frequency. So in total across this region, we found 60 individuals in Genomics England that had one of these variants. It was about 0.7% of the undiagnosed NDD programs. Now, one thing that is incredibly difficult is to actually put an accurate frequency on this. So we do not have a completely unselected cohort that have not had any prior testing that have whole genome sequencing, because the majority of cohorts that have been built to date have an amount of pre-testing that allowed them entry into the cohort. And that's true of the 100,000 Genomes Project. Despite the fact that I said now in the UK, we're doing it as a first tier test, that's the data that's coming through now. In the 100,000 Genomes Project, there was a certain list of genes that had to be tested before they were entered into the project. But if we use from this paper that I used on that very first slide, which had a 40% diagnostic rate in the DDD study, and assume that there is a 40% diagnostic rate upstream of defining this undiagnosed NDD cohort, this would estimate that variants in this gene cause about 0.4% of all neurodevelopmental disorders. That sounds like quite a small number, but I'm just going to flash up 2 genes here. So these are the genes that are the most commonly mutated in this paper, in this cohort from the DDD project, which is, I think, about 1,500. Oh, no. I can't remember how many individuals. Anyway, these 3 genes are the kind of big hitters, and they're very well-known NDD genes. And as you can see, the frequency is not too dissimilar. But the thing that's incredibly striking is every single one of these genes is in excess of 2,300 amino acids long. So they're huge. And we're talking about an 18 base pair region of 141 base pair RNA. Pretty cool. So what is this RNA doing? So this is a small nuclear RNA, which means it's a component of the spliceosome. So it encodes the U4 snRNA, which is shown here in pink, just right in the center of this major spliceosome complex, and it's tightly wound to another snRNA, U6. So this is a figure about splicing, and I know that Marco knows a lot more about splicing than me, but these snRNA genes are responsible for recognizing the splice sites. So particularly U4 is important at the 5' end. So U1 recognizes the 5' splice site, and it recruits this what's called tri-SNP, which includes U4 and U6 and also U5. And this U6, so U4 and U6 have to be unwound from each other in order for U6 to receive the 5' splice site and splicing to be activated. So just to look at that a little bit closer, we're going to zoom in once again. Here we have in teal this U4 snRNA, and in grey, part of the U6 RNA as it is bound to U4. And underlined here is this 18 base pair region where we find these mutations, with the position of this single nucleotide variants in orange and the position of the insertions marked by arrows. And as you can see, they cluster to two distinct regions here. One is in this STEM3 region, and this other region here on the cryo-EM structure from the spliceosome is part of this quasi-pseudoknot or T-loop structure. And what these two structures are thought to collectively do is to hold the beautifully named U6 agar-agar sequence in the correct position to receive the 5' splice site. So we decided to look into the RNA sequencing data that we had for 5 of these different individuals with RNU4-2 variants to see if we could actually see any defect in splicing. And this was done by Jenny Lord, who used to be down in Southampton with Diana Borale, but now is a lecturer in Sheffield, and Ruby Dawes, who's a postdoc in my team. And what we saw is if we specifically look at the 5' splice sites of these so we specifically look at significant events that corresponded to a differential usage of a 5' splice site. So we move away from using the canonical splice site to using a cryptic site nearby. We saw an increase in these events that were significant in the cases compared to either a whole bunch of controls or 10 controls matched 2 per case on age and ethnicity. And what was actually quite striking about these events was actually they were quite often shared events. We saw the same events happening in different individuals. So it looked like it was a systematic shift in usage of these splice sites. So on the right here, I'm showing you if we randomly sample 5 controls from the 378, how many times we see the same event, and basically 0. So we did this 10,000 times, basically always 0. Whereas in the individuals with RNU4-2 variants, we saw 11 different events that were in common. So this looks like a systematic change in 5' splice site usage. So we looked into this a little bit more detail. So here what I'm showing you is a sequence motif where on the top here is, well, it's on the bottom actually. On the bottom here is the consensus, is the sequence at the 5' splice sites that are decreased in usage systematically across these patients. And this basically matches the canonical 5' splice site. And on the top, what I'm showing you is the sequence at the sites that are increased in usage, these cryptic sites that we see increased in these RNU4-2 individuals. And what we can see is a change away from consensus at the +3, +4, and +5 positions, where we have an increase in T and an increase in C at 4 and 5. And what we found really interesting is that these 3 positions are the exact 3 positions of the 5' splice site that directly pair with the U6 agar-agar sequence, supporting the mechanism that this is disrupting, these mutations are disrupting that interaction between the U6 agar-agar sequence and the 5' splice site, which is quite a cool way of circulating back and looking at mechanism. So I'm just going to hit on 3 different points that we've had a lot of discussion around since we made this finding. And this is all very new, so still a lot of unknowns in this area. So one thing that was mentioned very quickly when we first put the preprint up of this back in April was, can we actually identify these variants ever in exome sequencing? And the reason why this is important is the vast majority of undiagnosed individuals across the world have exome sequencing. Genome sequencing is still very expensive. And there was the wonderful Francoise on Twitter, actually the day after I think we posted the preprint, had already written a bit of code to look through their entire cohort and look to see if they could find anybody with even a single read at the primary insertion position that was carrying the insertion. And lo and behold, you can actually find individuals that have 1 or 2 reads containing the insertion, and some of these have then been validated and they have the variant. However, you need rather a lot of luck. So there are actually 22 individuals that are in both Genomics England and also in the Deciphering Developmental Disorders, or DDD cohort. DDD cohort has exome sequencing, Genomics England has genome sequencing. So this allows us to actually compare and see if we can identify any of those individuals from the DDD exome sequencing data. None of them have a single read that overlaps the position. So whilst you could get very lucky, it's definitely not a very sensitive approach to try and identify these variants. So we really do need to be doing targeted sequencing or genomes to try and find these variants. So one of the other things we thought about is, why on earth is this variant so recurrent? So as I said at the beginning, I wanted to chuck this result away. I thought this was crazy. This is not what we were expecting to find. So why do we see this in so many individuals? Well, one thing to note is that overall we find a huge mutation density across the RNU4-2 gene, and actually all different snRNAs have a similar phenotype. So what I'm showing you here is in gray, this histogram here is the median proportion of all possible single nucleotide variants. So every single different single base change, each position across 141 base pair chunks. So 1,000 random 141 base pair chunks on chromosome 22, which is the chromosome that this gene exists on. And we see, on average, 13% of all possible SNVs observed. In RNU4-2, it's 76%. So they're hugely mutation dense. There's something driving this high level of mutation. And there is also a second gene right next to RNU4-2, which also encodes U4 on the same chromosome, and it also has this very high mutation rate. There was a paper recently that actually proposed that this high mutation rate might be linked to transcription of these genes by polymerase 3 rather than polymerase 2. And these kind of RNU genes and tRNA genes that are pol3 transcribed have this much higher mutation density. One of the other ideas I thought, although this is completely unproven, it's entirely guesswork, is that maybe because this region forms secondary structure as a single-stranded RNA, maybe when it's unwound during replication, it also forms a secondary structure which drives indel formation through some kind of replication slippage. As I say, complete guesswork. Another idea is germline selection. I also said right at the start of this talk that some of the mutations we do find that are highly recurrent are such because they are selected in the germline. But the only place where this has been studied and shown is when this is selection in sperm. So when you have mutations that confer a selective advantage to sperm, they're called selfish mutations. They occur far more frequently than we would think. And this leads to a higher rate of these in often severe developmental disorders. But rather interestingly, when we tried to phase these variants to determine which allele, whether it's the paternal or maternal allele that the de novo variants arose on, every single one of them that we could decipher were maternal. Now, if you're not used to this, you might not realize that that's such a striking result, but actually what we expect is 80% of these mutations to be derived on the paternal allele. So the complete absence of any paternal allele in mutations is very interesting. It potentially hints at the fact that these might be lethal in sperm if this gene is incredibly important during spermatogenesis. But this is all stuff that we need to work on a bit further. So the final one is, if this gene is so recurrent, why on earth has it been undiscovered for so long? Now, there are other snRNA genes that have been shown to be involved in disease. These are RNU4 attack and RNU12. They are both components of the minor spliceosome, which is associated with splicing a very small proportion of, I think, about 400 introns across the human genome, whereas the major spliceosome is involved in everything else. And these genes cause a recessive phenotype. And looking at recessive, we can do things like tracking alleles in families, and we can look at related consanguineous individuals, and we're more likely to identify these disorders. Whereas because we're looking at a de novo phenotype, this is much harder to identify in families or smaller data sets. I think I went to the third one first, anyway. And there is also, as I said earlier, no coverage of this in exome sequencing data, so this has not been identified to date. And also, the other thing is that the people have actually looked selectively at these genes. So Matt Hills' group at the Sanger Institute actually looked in developmental disorders, specifically in Genomics England, at snRNA genes. But they were using mutational models that look purely at single nucleotide variants, which meant they missed the signal. So this kind of begs the question of how many others of these out there are out there. So are we going to find other genes with such a striking role in neurodevelopmental disorders? And lo and behold, there's another team that actually also discovered RNU4-2 at a very similar time to us, who've just put out another paper talking about a different gene. Actually, interestingly, is thought to be a pseudogene in which they find variants at much lower frequency, but that might also cause severe neurodevelopmental disorders. So we think there's a lot more to be found in this space. So one of the things that's been most fun for me, this is the closest I've got to interacting specifically with families for anything that we've discovered, and it's definitely the most impact I've had in my research. This is a screenshot of one of the parents very quickly set up a Facebook group, and this is now has been growing. Every time I use this slide, I have to update it, and now has 154 members. And one of the things that's really important about a diagnosis that's often understated is the importance of these families connecting with each other. So imagine if you have a 3-year-old, you know they have this severe disorder, you've got no idea how long they're going to live, how that disorder is going to progress. But if you suddenly meet somebody who's got a 20-year-old, you suddenly know what puberty looks like, and you know that your child has a future, and that's incredibly powerful. The name Renew syndrome was actually also derived by the families. Omim tried to call it something terrible like Ned Hather, based on some of the symptoms which the patients hate. And instead, we came up with this wonderful Renew syndrome because one of the parents described it as renewing their family because they now have hope for the future, which I thought was really beautiful. We're also been working with the charity Unique to try and connect families and do Q&As basically. And write information guides for older families that are being diagnosed. And this is just a wonderful quote from one of the families that I really love about kind of their diagnosis. And I really like this bit here, right? So this is where data meets real life. As researchers, we get very kind of in the data. And sometimes we get to kind of think, oh, this is the impact that we're having. And this is where this Renew name came from. We like to refer to this RNU4-2 as Renew, as this family is being renewed by this new information and hope for the future. Okay, so that was my first story. I'm going to pause and see if anybody has any questions before I move on any further. We're going to my favorite regions of the genome, which are untranslated regions, or UTRs. They're mostly ignored by a lot of people, but I think they are fascinating. So what are they? So they are the regions directly up and downstream of the protein coding sequence of a protein coding gene. And they have incredibly important roles in regulating the stability of the RNA, the rate at which it's actually translated into protein, and where the RNA is localized to within the cell. And this regulation is mediated by a series of different sequence motifs and secondary structures, and the binding of these regions to external factors, RNA binding proteins and microRNAs. My favorite regulatory region within my favorite region of the genome are these elements called upstream open reading frames, or UTRs. So these are small regions that look like mini protein coding sequences. They have a start codon and a stop codon, and their role is to disrupt the scanning ribosome. So during translational initiation, the ribosome, the small subunit of the ribosome binds the 5' cap of the RNA, and it scans along until it finds the start codon of the protein coding sequence, at which time it translates. But these UTRs, the ribosome can actually translate this UTR on its way to translating the protein coding sequence, and they regulate the ribosome flow. And there's a study that showed that they confer around 30 to 80% reduction in protein levels. And crucially, they're not rare. They actually are predicted to occur in around half of all protein coding genes. So they're naturally occurring negative regulators of translation. And just as a bit of terminology, I'm going to refer to these start sites as upstream AUGs or UAUGs. So we can detect these UTRs using a protocol called ribosome profiling, or RiboSeq. And this is an approach by which the RNA is digested and the regions that are actively bound by a ribosome are protected. And so we get these what we call ribosome footprints, which are actually a defined size when they're protected by the ribosome. And then we sequence these, and we can look to see where these ribosomes were sat on the RNA. And then, because some ribosomes can just be sat there for no apparent reason, we then have to check whether this looks like active translation. So ribosomes actually move along in 3 base pair chunks as they're translating each codon. So we can then look to see if we see this 3 nucleotide periodicity in the riboseq reads. And then we look to see if we get to a stop codon and we see no more riboseq reads. And these are our 2 main filters, this periodicity and then this ribosome drop-off. So when we look at these data, and also when we look at predictions, there are actually 3 different types of upstream start codons, or 3 or 4 types. So either we can encode a UTR, which I already introduced, where we have a stop codon in frame with a start codon still within the 5' UTR. We have these very weird elements that have been discovered more recently, which are called start stops, where you have a start codon immediately followed by a stop codon. And these are thought to be ribosome confusion elements or pausing elements, because here the ribosome is simultaneously trying to initiate and terminate translation. So it kind of confuses them. Or we have these occasions where we have a start codon and no in-frame stop codon within the UTR. So here we have, if the ribosome started translating here, it would bypass the start codon of the protein coding sequence. The ribosome wouldn't be available to translate the normal coding sequence. Or sometimes we have these ones that are in frame, so you just translate an extended coding sequence. So there are multiple types of different upstream AEGs that can have different effects on protein translation, but in the majority is that they are negative regulators of translation. So we've done a lot of work on this, looking at variants that create new ones of these upstream AEGs. We show that they are actually under this paper here, we show that they are under very strong negative selection, equivalent to protein coding missense variants, so amino acid change variants. So they're under very strong negative selection. And more recently, and I'll come back to this paper in a minute, we identified variants that create these early start codons, specifically in a gene called MEF2C, that lead to severe developmental disorders. So they can cause a very large effect. So we found these variants that have a single mechanism where we predicted that creating an early start codon might be deleterious. But what we wanted to then ask is how frequently do any variants across the UTR with different mechanisms cause a rare disease? And this is work by a superstar postdoc, Alex Martin-Geary, and it's actually also available as a preprint. So here we looked at specific regions of interest. So we ignored the tiny little grey boxes in the middle that everybody thinks is really interesting, the protein coding regions, and instead we focused on the UTRs. So the exons of both the 5' and the 3' UTR shown here in purple, the introns shown here in blue, and also an upstream promoter region predicted here from ENCODE dataset. We then kind of did this really comprehensive bioinformatics annotation of variants in each specific region by hypothesized effects that we of these variants. So in variants in the promoter, we annotated with variants that would likely disrupt transcription factor binding, for example. In the 5' UTR, we had annotated variants that would disrupt the so-called Kozak consensus sequence near the protein coding start site. This is the sequence that dictates the level of translation from that start site. We looked at variants within predicted internal ribosome entry sites. These are sites where the ribosome can bypass having to bind at the 5' cap of the RNA and instead jump in halfway through. We used a tool that we previously created to annotate these start codon creating variants and also variants that disrupt existing upstream open reading frames. And we used Splice AI to annotate anything that was disrupting splicing. In the 3' UTR, we looked at variants that disrupt polyadenylation, that disrupt binding of either RNA binding proteins or microRNAs, or that disrupt these so-called downstream open reading frames, which are also small translated regions that somehow exist in 3' UTRs. Don't quite know why. And across all exons and introns, we use Splice AI. And then finally, because in this approach, we're hypothesizing what effect a variant might have, we thought, well, we might be missing something because we don't know everything about 5' UTRs. So we also annotated variants that were strongly conserved using CAD scores and PhyloP. Okay. So what we did is we took a set of 8,000 probans, individuals within Genomics England that didn't have a diagnosis. We identified then rare de novo variants. So again, these are the variants that specifically exist in an affected child, but not in either unaffected parent. The reason we do this is these de novo variants have a much higher prior probability of being pathogenic in this scenario where the parents are unaffected. We then looked at our annotated regions that I showed to you on the last slide. In a set of genes that are flagged as Genomics England as green genes in their panel app program. These are genes that basically are a known cause of a patient's phenotype. So these genes will already have been screened for protein coding variants. Then we're just saying, but actually in a known gene, can we find a regulatory variant that causes disease? We then did our region-specific filtering that I showed on our last slide. And the key result here is that from 8,000, we ended up with 11 variants in 11 probans. So our hit rate here is quite low. So of these 11 candidate variants, 4 of these we call positive controls. These are things that we already knew about. So these are 2 variants in MEF2C, one variant in MEF2C, 2 variants in PAC6, and 1 variant in RPL26, all of which had already been identified as pathogenic in Genomics England. I told you I'd come back to this paper, and this is because I think this variant has a very cool effect. So this variant in MEF2C was one that we found as part of this paper that I mentioned earlier. So MEF2C is from the MEF2, the MEF2 family of transcription factors. So here is a structure of MEF2A, the N-terminus of which is identical to the N-terminus of MEF2C. It binds as a dimer, one in orange here and one in teal, and the N-terminus binds directly into the DNA for this transcription factor to function. And what we actually identified, I have missed off a previous slide, which have helped here. What we actually identified here were variants that create a new start codon that extended the N-terminus of the protein coding sequence. And here what they did basically is added 3 and 9 amino acids respectively. So one of these we found in gel and one of them we found in the DDD cohort. And initially we were confused as to why this would have a large effect. But actually when we looked at this structure, we saw that adding a small amount of sequence to the 5' end so the N-terminus of this protein would completely disrupt DNA binding. So we tested this in an assay. We cloned mutant and wild-type 5' UTR upstream with MEF2C, and then had a luciferase reporter gene with MEF2C binding sites upstream. And we saw that compared to the wild-type, where we see this large transactivation, so expression of luciferase, these mutant 5' UTRs dramatically reduce the transactivation. So I think this is a very cool mechanism that I like to show because it's a very protein coding effect of a variant that's found in the 5' UTR. So there are ways in which UTR variants can actually affect the function of a protein. So what else did we find? Of the remaining variants, the wonderful Alex Blakes did a clinical review, and 5 of these we thought were likely new diagnoses. One of the interesting notes here is that we have a very high hit rate here. So we actually think 9 of the 11 that we prioritized are likely diagnostic. And this is very important because in a clinical setting, we can't just say to clinical scientists, you now have to interpret every variant in non-coding regions, because that's going to be a lot of work. But if we have ways in which we can filter these down in a very specific manner to decrease the amount of interpretation they have to do, this is incredibly useful for clinical teams. So what do these variants look like? One of these variants was a variant that created a new upstream AUG. So like I described earlier, that was out of frame with the coding sequence. So here, if the ribosome starts translating early, it won't be available to translate the protein coding sequence and will cause a loss of function. A second variant was in NICBL, and this was a variant just upstream of the final acceptor sites of the exon, which is partly UTR and partly coding sequence, so it contains the start site. And this was created in AG within the so-called AG exclusion zone, where basically this is deleterious, and if this would cause exon skipping, it would remove the start codon of the protein. However, I would say that we don't have RNA sequencing data to prove that this is the effect. The third one was within a, a region that was annotated in one transcript as 3' UTR, but actually is likely a protein coding effect, because in another transcript, this is a splicing variant within the coding sequence of this gene GNAS. And this, by Jenny Lord, we showed in RNA sequencing data, reduced expression and altered splicing of this GNAS gene. Thirdly, we have an interesting variant that we identified. Fourthly, I can't count. An interesting variant in SETB5. And this is another splicing variant identified again by a very high splice AI score. But it exists right next to this upstream donor site. So how this is mediating disease, maybe possibly through some intron inclusion that just changes the 5' UTR sequence, is currently unknown. But there was actually a second variant at the same site that also an individual with a similar phenotype. And we showed using a methylation assay in collaboration with Siv Banker that these variants in these black dots here had a methylation signature very similar to other individuals with SETD5 loss of function variants, rather than clustering with individuals with control individuals. And finally, just an example to show that we're still very far off being able to interpret a large number of variants that are in non-coding regions. We found a variant that looks like a good phenotypic match in ZBTB18 that is 13 base pairs upstream of the transcription start site. It was prioritized just through being conserved, but we have no current hypothesis of how this is causing disease. So just some rough conclusions from this work. So we found a diagnosis for a very small proportion of individuals. So including the second SETB5, we then get to 10 individuals out of that 8,000. This is a very small number. However, this is kind of likely underestimating our diagnostic rate, because we had this very strict filtering approach to make sure that we were being very specific. And we're also very lax on the definition of undiagnosed. There's a proportion of these 8,000 that likely have a protein coding diagnosis. However, the main conclusion is that despite these caveats, we're unlikely to find a huge fraction of undiagnosed patients with variants in UTRs. But where does this leave us? We've learned a lot about UTR regulation. Is there a way in which we can actually use this moving forward? So just a reminder, so upstream open reading frames, my favorite regulatory elements, are these negative regulators of translation that exist within the 5' UTRs for around half of all protein coding genes. So an idea here is, in the therapies space, can we actually modify UTR regulation as a therapeutic approach? So imagine a scenario here where we have two alleles in a healthy individual that each produce 2 units of functional protein. In the disease state, we have a loss of function variant. That means we only have one functioning allele, and 2 units of functional protein is not sufficient. But what happens if we could remove a negative regulatory element in the wild type allele and upregulate protein expression from that wild type allele to a sufficient level to recapitulate natural protein levels and treat a disease? So there is a couple of papers in the literature that say that we can actually target antisense oligonucleotides directly to UTRs to upregulate downstream protein expression. There is also a lot of people that have tried to recapitulate this and have failed. And there's one paper that actually said that this, specifically on the gene, one of the genes that was done by this team, that they could not replicate their results. So directly targeting UTRs does not seem like a credible approach. And I think this makes sense. You're putting a steric block to a scanning ribosome by putting an ASO on a UTR. But one thing we do know works is splice switching ASOs. So there are multiple different therapies in development. This is just a couple of examples: the approved SMA therapy and also the SCN1A Dravet syndrome therapy. It's in clinical trials that use splice switching ASOs. So can we use splice switching ASOs in a UTR context? So we can in some genes where we have this particular structure. So here the exons are my boxes, and the introns are shown by these lines. So here if we have a UTR, or in fact just the start codon of a UTR on a central exon, could we use a splice switching ASO to exclude that exon and upregulate downstream protein levels? So this is a project that's been being done by Eloise Wells, who's a wonderful PhD student in the team. She's used a set of representative transcripts from the main project and found that there are about nearly 1,600 transcripts that have at least one central, potentially skippable UTR exon. We then use ribosome profiling or riboseq data to identify UOF start codons in these central exons. So we have a whole bunch of different riboseq data that is available. Some of this is from primary cells and tissues, but a lot of it is from cell lines. So there's some caveat that this is not exhaustive at this point. But we put a lot of effort into trying to coordinate these data and identify small open reading frames or UOFs more generally across these data set. And one thing to note is that although the majority of these start from an ATG, translation can actually start from another codon, often NTGs. So not all of these are ATG start codons. There are some that are most often CTGs. But if we correlate these genes with these potential skippable exons with genes that have a UOF start codon on that exon, we get about 800, well, just shy of 900 genes. And if we correlate this with the list of known neurodevelopmental disorder genes as a starting place, we find we have 66. So there's potentially a few tractable genes where this approach might work. So this is where the wonderful Laura and Marco came in. We approached them to help us with some validation of this. We tried 3 different examples, one of which is GRIN2B. And what we've done here is we've cloned different 5' UTRs upstream of the luciferase reporter. We have the wild type UTR, we have the UTR missing the central exon, and we have the UTR with the central exon, but missing, in this case, multiple ATGs that exist within this central exon. And here we can quite nicely show that removing the central exon upregulates protein expression, and this effect can also be recapitulated by mutating the ATG. So it looks like it's a UOF-driven effect. One of the caveats here, though, although this looks really neat and we got very excited by it, GRIN2B has a severe neurodevelopmental phenotype associated with gain of function as well as loss of function. So a 7- or 8-fold upregulation is possibly too good. But it shows that the approach works. So just on this project, we've shown that removing a central 5' exon can dramatically increase protein translation, well, luciferase translation. Although we need to kind of take this into broader context than a natural RNA environment. So we need to do some ASO testing for both GRIN2B and other targets. Our initial list was NDD genes, but there's no reason that has to be limited to NDD. And we're also exploring multiple different other mechanisms by which we can perturb UTR regulation as a therapy. So finally, just to acknowledge my absolutely wonderful team who are shown here and a whole bunch of different thunders. And thank you very much for listening.